and we move on to the next speaker, which will be Robson Tramontina from Institute of Biology from Unicamp, Brazil.
Okay. Are you guys able to see my presentation?
Yes, we are.
I'm not sure if I can share my video screen. I'm not allowed to do that. So if someone could please open the video for me from the staff.
To open what? Your video? You can't.
I can appear in the screen. It's blocked.
Oh, sorry. It's,
Okay,
probably.
thank you.
Okay.
Right. So are you guys able to see
Yes, we are.
Perfect. So thank you very much. Last but not least, I would like to thank you, the previous talkers, Adibari and Igor. It was a delightful presentation. It's a pleasure also to be here talking, especially in front of Igor. I have been reading a lot of articles. I know many former students from you, so I have been collaborating with them as well here at Unicamp. So it's quite exciting to be sharing this table with you guys. So my name is Robson Tramontina. I am from the University of Campinas, Unicamp. My supervisor is Fabio Esquina. It's an enzymologist here in Brazil. This work was done partially at the University of Campinas in the Biology Institute, in the Faculty of Food Engineering, and the School of Chemistry as well, but also during an interchange period in the Manchester Institute of Biotechnology. The title today, I'm going to talk today about integrated production of high-value aromatic alcohols directly from lignocellulosic biomass. I also work with biomass detoxification and production of enzyme for development of IVD diagnosis purposes, but today this is going to be my focus. This is quite fun by using synthetic biology approach to add value to the biomass. So I decided to put a summary here so you guys can understand better what I'm going to talk during the presentation. So lucky me, I'm not going to have to go extend myself explaining what's the lignocellulosic biorefinery, but I'm going to focus on ferulic acid as a bio-based platform, so a ferulic acid biorefinery, and explaining also about the product, the target product I developed, which is coniferol. It's a high-value molecule. The use of biocatalysts or enzymes for producing those molecules. I'm going to talk about two enzymes by especially taking as example the carboxylic acid reductase, the CARs, and the aldoketon reductase, the AKRs. How I set up this research to develop these goals of producing coniferol, the in vitro development of these cascades, the in vivo development of ice strain that was capable to produce those components, the use of real biomasses for the production of those components, the conclusion of this work, and the further studies which is related with design of experiments and integration in a real scenario by using sugarcane straw to produce lots of components besides coniferol and ethanol. So there you go. As Igor mentioned before, the lignocellulosic biorefinery, it's the best alternative to replace the phosphate-based refinery, which is a finite and not very eco-friendly manner to produce fuels, chemicals, and energy. So we can say that it's the ultimate recycling of those feedstocks. Generally, it's produced by using agro-industrial residues, so it doesn't compete with food production, for example. It's very available, especially here in Brazil. We have loads, piles of those residues to be produced, and we should produce much things with them besides just burning them to generate energy. And of course, all those concepts of developing a biorefinery should take into account the social impacts, the economic impacts, and the environmental impacts that that approach is related. I would like also to mention, that we could produce lots of things besides ethanol or beyond ethanol in the biorefinery. Any sugar-based platform or phenolic-based platform should be developed for the production of a wide range of chemicals. Today, I'm going to focus on the fine chemicals, which has a smaller market than fuels, but they have a high value price, so it's worth sometimes producing them, right? So yeah, talking especially about ferulic acid. Ferulic acid is present in the hemicellulosic part of the biomass, especially on arabinoxylanes, which are present on sugarcane bagasse, for example. They are decorating hemicellulose, and they are responsible for cross-linking the hemicellulose and the lignin. So they have this main goal of structural protection of those biomass. They have also other roles in the plant. And ferulic acid is actually a phenolic molecule which has lots of interesting chemical bonds and chemical properties that could be used to generate a wide range of chemicals. Ferulic acid is present around 2% in the biomass by the total weight. So if you take a kilo of biomass, you're going to have a reasonable amount of ferulic acid to be extracted and is isolated. And one of those interesting assets from those ferulic acid is that there are some types of enzymes that are capable to hydrolyze their ester bonds that binds ferulic acid into the biomass, so releasing that ferulic acid into a soluble molecule. Those enzymes are called ferulyl esterases. So taking account the ferulic acid biorefinery, if you could be able to depolymerase, release that ferulic acid and perform a double reduction into the carboxylic group in that component, we are able to produce coniferol. And coniferol, it's a really interesting molecule. Besides its prompt use in the industry, especially by cosmetic industry, we have, it's a building block for a generation of a wide range of different molecules, especially fragrances such as coniferol acetate, which is the petunia accent, isoalginol, also it's another essence, and pynoresinol, which is a medicine used for hypoglycemic activity. So it's a really interesting target to be produced. But actually, coniferol is a high value molecule present in the market, has lots of challenges to be chemically synthesized because it's very sensible to oxidation of this double bond that it has in the propanil chain of that. So it would be desirable if you should develop a sustainable and green alternative to produce those molecules. And one way of producing coniferol from ferulic acid is by reducing the component using biocatalysts. For example, the carboxylic acid reductases. Carboxylic acid reductases are quite complex and big enzymes. There is not very known function in some microorganisms, might be related with protein biosynthesis, but uses ATP and ADPH to reduce those components, those acids into their respective aldehydes. Another interesting enzymes, and especially an enzyme that we developed and studied here in our laboratory, is the aldoketoreductases, especially the endogenous termite aldoketoreductases. It's a superfamily of enzymes capable to reduce aldehydes into alcohols. It's related to loads of functions on different microorganisms and pluricellular organisms as well, but mostly importantly, they are related with detoxification of, xenobiotics in the organisms. Especially in the termite, we found out that this enzyme was able to was present during the termite diets based on a lignin, which is a phenolic component of biomass. So we characterized that enzyme, and we found out that this enzyme was also present in the termite gut. This enzyme was able to detoxify hemicellulosic hydrolysates. So we would increase the fermentation rates by yeasts, but also was able to enhance the saccharification of biomass by producing reactive oxygen species. So it's a very interesting biocatalyst for biorefinery applications. So basically on that study that I'm proposing here, we wanted to go from pure lignocellulose, developing a biodegradation strategy by using an enzyme called SHINZ. SHINZ, it's a silanase chimeric enzyme coupled with a ferroesterase. So that enzyme is capable to release ferulic acid from the biomass. So ferulic acid is now soluble in the media. And we were able to develop a strain harboring CARs and aldoketo reductases to produce coniferol. So if you take into account the cost of these biomass feedstocks, it's around 7 cents a kilo, 1 cent a kilo of that biomass. And if you could produce coniferol from that, it's a high valuable product, around $360 a gram. So it's very worth, you know, trying to produce from natural sources, that component. So our first results here was to evaluate different CARs from different organisms in order to select the best biocatalyst for ferulic acid release, biotransformation, and coupling that reaction with the aldoketo reductase from the termite that already presented the activity against coniferol aldehyde. So after in vitro studies and, you know, LCMS analysis, we were able to find that Nocardia ioensis was the best biocatalyst so far, and was compatible, was really good in synergism with aldoketo reductase. So we decided to further develop this strain, these biocatalysts. Here, we already developed the strain, so we built a plasmid containing the aldoketo reductase from the termites, and comparing to a cinnamyl alkyl dehydrogenase, which is an enzyme already employed, was before employed in the reduction of cinnamyl aldehydes, and the Nocardia CAR. What we see here, it's because we used a strain of E. coli called rare strain. It's a knockout strain that doesn't have any endogenous aldoketo reductases or alcohol dehydrogenases. So we were able to evaluate only the heterologous system to aldehyde reduction inside the cell. And we were able to prove that the termite aldoketo reductase was actually a little bit better than the enzyme already used in the literature. So we decided to keep this enzyme in our heterologous system to go beyond. Basically here, we went further by expressing those system NBL21 E. coli strain. That strain harbors endogenous aldoketo reductases and ADAs as well, and compare it actually with the rare strains. The Nocardia strain actually without the ACAR and with the ACAR. And the take-home message here is basically that indeed Nocardia strain was the best option for the production of coniferol aldehyde here. The heterologous system actually worked pretty well on the conversion, but was not very efficient. Neither the endogenous systems alone as well here. But when we combine the endogenous with the heterologous aldehyde reduction system, it was quite efficient. So that was the best strain. Nocardia CAR in BL21 with the aldoketo reductase. We also evaluated the media composition, and we observed that terrific broth was the best option because it's a very rich media containing carbon sources, nitrogen sources, and was buffered solution as well. Could impair in the could enable a higher density of cells in this system. And the reaction was also quite fast. After 2 hours or 10 hours here, we have complete reduction of the ferulic acid and production of the coniferol alcohol. So the system was quite efficient and was comparing to the literature. They have another strategies to produce coniferol by using 4 or 5 coupling enzyme system, different from this system I developed here. It was the best so far in comparison to the literature. We are also able to see and compare another phenolic acids from natural sources or synthetic sources. And we were able to see here, the take-home message is that the system was compatible with loads of different cinnamic acids. We have mostly of alcohol production from them. Some of them, we only were able to reach until the aldehyde. So the take-home message here is that this is a platform for production of phenolic alcohols coming from aldehydes or phenolic acids. So, and finally, we went to the use of real biomasses. Here we use it wheat straw, which is a biomass commonly available on UK. And we use it wax, which is a arabinoxide coming from the wheat straw. So it's a more concentrated source of ferulic acid. So we actually employed the use of Shinzi in this system, and we compare ferulic acid and coumaric acid released in comparison to the alkaline treatment here. And we could observe it that they were equivalent. So the biocatalyst here, Shinzi, was actually in that scale and was comparable to the alkaline treatment. So that was quite interesting. And after we performed this degradation step in this bioprocess, we applied the use of the strain, the PROB, I call here PROB strain, harboring aldoketo reductases and CARs, and that strain was capable to convert all the released ferulic acid from the media. So we have 100% conversion of those components here. And by performing a simple technical economical studies, we actually coming from the use of the media and antibiotics and feedstocks, we were able to increase by 74-fold value added if you would sell those coniferal coming from this process. So the conclusion here is that we developed a simple method for coniferal production. It has high purity, high yield conversion, so very efficient method. Most importantly, we developed that by using a plant biomass feedstock. So we use real biomasses to perform that using biocatalysts such as enzyme, those mentioned enzymes in ambient and expensive extraction conditions. So it's a really eco-friendly process to develop a fine chemical. And most importantly, we developed a platform to create further organisms for other chemicals. So the next steps are going to be related to synthetic biology approach to expand those toolbox and created further components in synthetic biology to sense the biomass, degrade that biomass, and convert that biomass into another molecules that we already have in mind. Another experiment we performed here, this is it's almost a finishing experiment. We performed a design of experiments and scale up of that previous developed process. So we were able to optimize ferulic acid conversion into coniferal by 2.2-fold increase if you develop an optimized condition that we saw, but also by adding a feed batch reaction by adding low amounts of ferulic acid and glycerol in the media during the time. So the strain was capable to increase the production of coniferol. It's very important to mention that ferulic acid and coniferol aldehyde, especially, and coniferol, they are actually xenobiotics. They are very toxic to cells. So it's not very easy to handle that during the cultivation, during the fermentation, because you can kill the cells by just not taking care of them. For example, not giving good aeration or adding too much ferulic acid. And the final step here, this is basically the last step in my thesis, is the integration of all those components, all those processes into a real biorefinery context. So we basically here, by starting from sugarcane straw, we were able to develop several bioprocessing, several unit operations here to produce molecules, different molecules coming from all the sources of biomass. By using the cellulose, the hemicellulose, and the phenolic component of the biomass, we were able to produce in different scenarios here, ethanol, xylo-oligosaccharides, and coniferol. And we performed here in 3 different scenarios. So by using the alkaline treatment to produce coniferol by alkaline extraction, saponification, but we also produce ethanol from the cellulosic component of those components. A biological treatment that were able to release both xylo-oligosaccharides and ferulic acid to produce a little bit of coniferol and xylo-oligosaccharide as well as ethanol, but was not very efficient. Remembering that there's a real industrial setup. So this biomass has a really big and thick mesh, was not very well, the setup was not like on workbench so far. So the efficiency of the biological treatment decreases a lot. But when you mix those treatments, you have high yields of coniferol production, high yields of xylo-oligosaccharides production, and big yields of ethanol production. Of course, we're still developing the technical and economical analysis of those streams, those scenarios, but we will be finishing this by the end of my PhD defense. So this just to show that it is possible to integrate all those components into a biorefinery context in order to maximize the profits in this area. That's going to be the future of the biorefinery.
The time is up now.
Okay. Yeah. Thank you very much. I appreciate your time. Thank you so much. And I would like to thank my supervisor, Fábio Esquina, my collaborators, and FAPESP for the scholarship.
Thank you, Robson, for a nice presentation. We're now open for questions. No questions from the audience?
There's one question from Igor.
Nobody is brave enough to ask, and I will ask Robson. Excellent work. I think it's very, very impressive. If I understood correctly, you did a 3-step transformation, right? Correct me if I'm wrong. And which one of these 3 steps was the least efficient? What is the problem with the transformation, basically? Can you comment on efficiencies of the ferulic acid production and then transformation into whatever was?
Oh yeah,
coniferol was advanced, of course. Yeah.
Well, if you're talking about the last topic, the scenarios I created with real biomasses, direct biological treatment with recombinant enzymes for Nicolai was not very efficient because you have to produce a lot of enzymes to treat a kilo of biomass. And sugarcane straw is not so rich in ferulic acid such as wheat straw or rice bran, for example. So we have less ferulic acid and it's more recalcitrant biomass. And as I decided to use a really thick, a really industrial feedstock, the release of ferulic acid was not very good as in the previous work I did in the Manchester University. So the biological itself wasn't good. So if you would produce coniferol in real scenarios, we would have to perform, at least in that moment, alkali extraction. And we could also use enzymes after that, but focus on xylo-oligosaccharides, because once you perform the saponification, you don't need to release any further ferulic acid. So yes, the mixed scenario was the best, but it's expensive and we still have to evaluate the price of producing those components by doing a lot of unit operations, you know.
Cool. Thank you.
Any more question? So if there are not any questions, I'd like to thank you all the speakers for participating in this webinar, and it's been excellent to have you here. And I'd like to take the opportunity to make a little bit of a announcement of our next webinar that's going to take place in 2 weeks, June 23rd, at the same place and time. And you can just follow the same links. And I would like also to remind you all, and for those that came later, that we have the opportunity for application for fellowships to attend the UPAB meeting. The deadline is June 30, so at the end of the month. So all students and young and postdocs can apply for these fellowships, and further instructions are available at the website shown in the screen. So people interested in applying should definitely go to the UPAB 2020 website and see how to apply for these fellowships. So I'd like to thank again everyone for attending, the speakers and the audience, and we look forward to meeting you again really soon in the upcoming webinars. So I think that now we should give a round of applause for all the speakers, and thank you again for participating with us in this activity.
Thank you for the opportunity.
Thank you.
Thank you again, and we look forward to see you again really soon.
Thank you. Bye.
Bye. Take care.
Thank you. Bye.
Thank you.